Now, in the meantime, while he'd been travelling back, his wife Alexandra, who was at the palace with the 5 children, had been placed under house arrest. And when Nicholas came back, he too was effectively told, "You're now a prisoner, you're under house arrest here."
In July 1918, deep in the Russian city of Ekaterinburg, the last Tsar of Russia, Nicholas II, his wife Alexandra, and their 5 children were executed, marking the violent end of the Romanov dynasty.
I think the picture that most people had of the Romanovs was of a very privileged family, but a family with which they could identify.
For decades, the truth about what happened to their remains remained buried under secrecy and conflicting reports. It was only years later that scientists and historians began to piece together the evidence, uncovering so many graves, examining different bones, and studying DNA to confirm the family's fate. But when fragments began to surface from a hidden forest grave, could these bones truly belong to Russia's fallen royals? The grave in the forest. In the summer of 1918, the last Russian imperial family left the Alexander Palace and was held under house arrest at the Ipatiev House in Ekaterinburg. The family included Tsar Nicholas II, Empress Alexandra Feodorovna, and their 5 children, Olga, Tatiana, Maria, Anastasia, and Alexei. On the night of July 16th to July 17th, 1918, officers led by Yakov Yurovsky executed the family and 4 attendants inside the Ipatiev house. The bodies were moved at night and buried in secret. For many years, the exact burial place remained a secret. In 1979, 2 men, Alexander Avdonin and Gely Ryabov, began a private search based on the memory they had, existing information, and a document called the Yurovsky File. The file, attributed to Yakov Yurovsky, described where the bodies were taken. Avdonin and Gely Ryabov found soil that seemed to have been dug, fragments of clothing, wooden boards used as markers, and pieces of bone near a dirt track known as the Old Koptyaki Road. The location sits in a pine grove on the outskirts of the city. The 2 men kept their work a secret for many years. The Soviet state discouraged private investigators who asked questions about the past. Avdonin feared political trouble and waited until the early 1990s, when the Soviet Union had begun to change. In 1991, he told regional officials, including Governor Eduard Rossel, about the site he had located in 1979. Officials authorized a formal excavation and invited specialists to examine the site. Archaeologists, forensic scientists, and anthropologists opened the shallow pit with care. The grave proved to contain multiple bodies and scattered remains. For them, the condition of the material presented a serious challenge. Soil, weather, and time had broken bones and worn away soft tissue. Some bones showed signs of burning and cutting. Investigators recorded the position of every fragment and removed small artifacts such as buttons, fragments of military uniform, and shards of glass. Conservators cleaned and cataloged each small object. Every item received a label recording where it was recovered. The labels recorded depth and position in the pit, so later analysts could trace the origin of each sample. Information from the field, photographs, and maps accompanied the physical collection. Officials placed bones and artifacts into sealed containers for transport to laboratories. Scientists and officials paid close attention to the chain of custody. The team documented who handled each sample, when transfers took place, and where items were stored. Officials moved material to secure laboratories in Ekaterinburg and later to other centers for analysis. The paperwork and seals mattered because the remains would face rigorous testing by private investigators in multiple countries. Anthropologists examined bones to estimate age at death, sex, and stature. Forensic pathologists looked for trauma that matched historical accounts of the killings. The fieldwork produced a matched set of physical and documentary evidence. Small items from the grave, such as a gold button and fragments of clothing, dated to the early 20th century and matched the era of the imperial household. Geneticists later targeted teeth and dense bone for DNA testing. Those elements often preserve genetic material better than other tissues. Laboratory technicians removed surface material that might hold modern contamination and then took samples from the inner parts of teeth and from the dense parts of long bones. Technicians used careful chemical methods to extract tiny fragments of ancient DNA. Early studies in the 1990s relied on mitochondrial DNA to test maternal relationships. Scientists compared sequences from the remains to living relatives who shared maternal lines. They also used nuclear DNA markers where possible. The combined approach brought both anatomical and genetic evidence into the picture. Public interest grew as researchers published their methods and results. Journal articles and news reports named the lead investigators and listed institutions that participated in the work. The presence of international partners added weight to the findings. British, American, and Russian laboratories shared data and repeated critical tests to reduce the risk of error. The discovery changed a long-standing public mystery into a testable scientific question. The field excavation moved the story from rumor and legend to physical evidence that scientists could examine. The careful excavation, the cataloging of objects, and the chain of custody all prepared the material for complex laboratory work. Officials later arranged a state burial for the remains that best matched the imperial family. On July 17, 1998, a public interment at the Peter and Paul Cathedral in St. Petersburg gave the family a formal place of rest in the city, where many members of the Romanov dynasty had been buried over centuries. Scientific study continued after the burial. Researchers kept samples and records so later teams could re-examine the material with improved methods. The grave in the forest started as a secret that only a few locals knew. The work of two men who recorded the site, careful excavation by specialists, and the transfer of material to secure laboratories turned those fragments of bone and fabric into evidence. Researchers kept original field files and photographs so future researchers could check what was done. Families of the victims and historians followed developments closely, hoping the science would answer long-standing questions about identity and public talk. But when scientists finally began testing the remains, no one expected the results to challenge history itself. What did they find? The first DNA tests that changed everything. Scientists opened the grave material and sent samples to laboratories in Russia and in Europe in the early 1990s. A team led by Peter Gill at the Forensic Science Service in Birmingham worked with Russian colleagues, including Pavel Ivanov at the Engelhardt Institute of Molecular Biology in Moscow. These scientists faced a difficult task. The bones had spent so many years under the ground. Soil, heat, moisture, and time had broken down DNA. The team needed careful lab work to find reliable genetic signals. Laboratory technicians chose to examine the teeth and dense parts of bone for testing. These parts protect DNA better than softer tissues. Technicians cleaned the outer surfaces, removed possible modern contaminants, and then powdered tiny sections to extract what remained of the genetic material. The chemistry used to pull DNA from bone then produced small fragments. Scientists read short stretches of genetic code and compared them to their living relatives. Two kinds of genetic tests proved most useful. First, mitochondrial DNA, which passes down the maternal line, offered a clear way to test maternal relationships. Second, short tandem repeat markers, which sit in the nuclear DNA, helped establish parent-child and sibling links. The combination of maternal and nuclear markers gave a fuller picture of family relationships than either method alone. For the maternal line, investigators used their relatives who were still living and could trace descent through women. Prince Philip, Duke of Edinburgh, agreed to donate a blood sample. He shared a maternal ancestor with Empress Alexandra Feodorovna, so his mitochondrial DNA could serve as a reference. The match between the mitochondrial DNA from the grave and Prince Philip strengthened the identification of the Tsarina and her daughters. For the male line, scientists compared nuclear markers to known Romanov relatives. Samples came from members of the Romanov family who lived in several countries. A living Romanov relative in the United States, Prince Andrew Romanov of San Francisco, provided material that helped confirm paternal relationships. Additional comparisons included sequences from Georgy Romanov, who showed a shared mitochondrial trait with the putative Tsar. The multiple comparisons reduced the chance that any match was a coincidence. The researchers repeated tests in separate laboratories. Private checks included running extractions from different bones, repeating sequences, and testing blank controls to watch for contamination. Staff wore protective clothing and used clean room practice when possible. These steps aimed to separate true ancient DNA signals from modern contamination that could come from the people who handled the samples. Early results published in scientific journals in 1993 and 1994 showed consistent patterns. Mitochondrial sequences from the female remains matched the reference sequence from Prince Philip. Nuclear markers indicated family relationships that fit the expected parents and children. The combined data pointed to a family group that included an adult male and an adult female with 5 child profiles. Not all experts accepted the findings without question. Some raised concerns about the limits of the early methods and about how samples had been stored before tests. Some of the researchers argued that the tests in the 1990s could not read long stretches of DNA, and that a damaged sample might produce misleading results if contamination was not controlled. The scientists anticipated these critiques and invited additional testing by other laboratories. Later studies returned to the same bone samples with improved technology. Newer methods could read more markers and recover more of the nuclear profile. Those later tests confirmed the initial conclusions and added strength to earlier identifications. The repeat studies showed that careful early work, when combined with improved later methods, could produce reliable results. The scientific effort drew on a range of disciplines. Anthropologists provided age estimates and injury analysis from the bones. Forensic pathologists compared marks on bone with historical accounts. Geneticists supplied sequence data and relationship estimates. The combined evidence made for a stronger case than any single source could provide. The bone fragments tested in the early work came from the teeth and dense thigh bones. These parts often protect small amounts of DNA. For each sample, laboratories ran duplicate extractions and repeated the readings to check for mistakes. When tests showed a match, other laboratories then tried the same samples. International partners compared their results to confirm the findings. Scientists also ran blank controls to watch for contamination from lab workers. Where any doubt remained, those investigators sought fresh samples and used different methods to seek the same genetic signal and verification. The 1993 to 1994 work did more than identify bones. It showed a way forward for modern forensics and historical cases where time and secrecy had hidden the truth. Journalists reported the names of the lead scientists and described how existing relatives provided reference samples. The identification reduced uncertainty about the grave group and created a path for more detailed study. Those next steps included searches for missing family members and more advanced genetic testing. The early tests had established the family unit that researchers would later complete with additional discovery. The science in the early 90s moved the Romanov story from just a word of mouth to documented evidence. Researchers kept samples and records, which allowed future investigators to check and expand the work with new methods and fresh comparisons. But if the grave was nearly complete, why did whispers persist about 2 missing children, and could they still be found? The missing children and the 2007 discovery. The first tests left open a difficult problem. Reports and witness accounts suggested that 2 of the Romanov children were not in the Koptyaki Road grave. Searchers kept looking for signs of missing remains because the historical record and family lists did not match the first set of bones. In 2007, a new search near the main burial area recovered a second cluster of human remains. The new deposit lay a short distance from the original pit beside the old Koptyaki Road. Field investigators found small bone fragments, burnt fragments, and scattered pieces that lay close to the surface. The condition of these remains differed from the deeper, undisturbed grave that had been opened in the early 1990s. Archaeologists and forensic specialists treated the new material with extra care. Some bones showed evidence of fire and cuttings, which suggested deliberate attempts to conceal the bodies. Investigators numbered each fragment, photographed every discovered body part, and recorded exact positions on field maps. Small artifacts appeared near the fragments, including buttons and textile fibers that matched early 20th century materials. The laboratory work focused on small, dense bones and teeth because such material often preserves genetic traces. Scientists removed modern surface contaminants before sampling internal material. Those internal samples produced degraded DNA fragments that required careful amplification. Technicians used short nuclear markers and mitochondrial markers to build a genetic profile from damaged material. Mitochondrial DNA clarified maternal descent. The laboratory compared sequences from the new remains to the maternal profiles already associated with the main grave. The match supported the conclusion that the fragments belonged to the imperial family. Nuclear markers then helped place the fragments within the family structure. The combined approach narrowed the possibilities to one male child and one female child. Skeletal and dental analysis provided age estimates for the child's remains. Anthropologists examined tooth formation, bone length, and growth plate closure to estimate how old each child had been at the time of their death. The estimates matched the ages of the two missing Romanov children. The male child matched the expected profile for Tsarevich Alexei, while the female child fit the age range for either Maria or Anastasia. Identifying which daughter the female fragments belonged to required careful cross-disciplinary work. The skeletal evidence alone could not name the child because Maria and Anastasia were close in age and in growth markers. The genetic profile showed the female was a daughter of Empress Alexandra Fyodorovna, which narrowed the options to the two sisters. Anthropological measures and dental records produced slight differences that researchers used to argue for one identification over the other. Different experts reached different conclusions about the daughter. Some anthropologists favored Maria based on bone metrics. Other analysts found that the overlap in measurements left room for Anastasia as an alternative. The genetic data confirmed family membership, but left a degree of ambiguity about the accurate name when the skeletal indicators overlapped. For the male child, the genetic and osteological evidence aligned more clearly. The DNA profile for the boy matched paternal markers expected for a son of Nicholas II. The bone size and tooth development also matched the recorded age of Tsarevich Alexei. Later genetic work would establish the medical condition associated with the male child, which supported the identification through a separate line of evidence. Investigators applied strict controls to prevent contamination and to verify results. Laboratories ran blank extractions, repeated tests on separate samples, and sent material to independent centers for confirmation. The multi-laboratory approach reduced the probability of error and increased confidence that the new fragments came from the imperial family. Specialists checked the soil and found charcoal pieces and traces of heat near some fragments. Burnt bone cracks and changes shape, which makes measurement harder. Experts used microscopes to look for tool marks and for heat effects. Those tests helped separate old damage from recent disturbance. They took pictures and scanned each piece before taking samples. Conservators cleaned items slowly and stored them in stable rooms to protect DNA. After laboratory work, scientists wrote reports that explained methods and controls. Colleagues in other countries read the reports and repeated key tests where they could. Peer review added confidence to the identifications. The pattern of burnt fragments and scattered bone agreed with eyewitness confessions about how the bodies had been moved. Forensic observations connected the physical evidence to written testimony and archival files. Those connections helped researchers set the new material in a clear historical context. Experts discussed results at scientific meetings and published articles. Those conversations let specialists check raw data and test methods. In the end, the 2007 recovery completed the set of remains at the Koptyaki Road site and allowed later teams to search the family DNA for medical details. But when researchers examined the Romanovs' DNA more closely, they uncovered something no one expected: a genetic secret hidden for generations. What was it? The gene that proved the royal disease. Doctors and historians long described the heir, Alexei Nikolaevich, as a boy who bled easily. Records from the imperial household indicate frequent bruises, heavy bleeding after injuries, and episodes that required urgent care. Medical researchers set out to test whether a genetic change could explain those reports. Scientists focused on the genes that control blood clotting. Blood clotting depends on a set of proteins that work together to stop bleeding. One key protein is factor IX. A defect in the gene that makes factor IX causes a condition called hemophilia B. The research teams looked specifically at the F9 gene to see whether samples from the remains carried a damaging change. Laboratories selected the best preserved samples for this work. Teeth and dense sections of long bones provide the best chance to find original DNA after many years underground. Technicians cleaned the outer surfaces, removed any possible modern contamination, and then took tiny amounts from the inner parts of the teeth and the dense bone. The chemical extraction produced highly fragmented DNA, so the investigators planned tests that could read short pieces of genetic code. Scientists used the polymerase chain reaction to amplify small stretches of DNA. The method copies short target segments many times, so researchers can read them. The tests targeted regions of the F9 gene that are known to affect protein production when they change. After amplification, researchers sequenced the short fragments to record the exact order of the DNA letters. The lab work found a specific change in the F9 gene. The mutation sits near a splice site and is described as IVS3-3A to G. Technical notation matters to scientists because it points to the exact place where the gene message can be altered. A splice site helps the cell cut and paste the gene message correctly. A change at that spot can make the cell produce less functional protein. Analysis showed that the male sample carried the mutation on his single X chromosome. Men have one X chromosome and one Y chromosome. When a mutation disables a critical gene on the single X, the effect shows fully because there is no second X copy to balance the defect. The pattern of the finding matched the clinical descriptions of Alexei as a boy with severe bleeding. Female members of the family can carry a single copy of the mutation without having severe disease because they have two X chromosomes. Tests on female samples indicated that Empress Alexandra Feodorovna carried the splice site mutation in one of her two X chromosomes. At least one daughter also carried the change. That pattern explains how the condition moved from mother to son in this family. Laboratories ran many controls to make sure the result was real. Investigators included blank samples that contained no bone to check for contamination during extraction. They repeated extractions from separate teeth and bones and sent prepared samples to other laboratories for private confirmation. Several independent groups reproduced the finding, which strengthens confidence in the result. The change in the F9 gene affects how cells assemble the message that tells the body how to build the factor IX protein. When splicing is disrupted, the cell may skip an important piece or include extra bits that do not belong. The net effect is reduced or faulty factor IX. Clinicians know that low factor IX levels produce bleeding that can be severe and life-threatening. Historical medical information from the Romanov household describes episodes that match severe factor IX deficiency. Accounts list internal bleeding and repeated crises after minor injuries. The genetic finding provides a molecular explanation for those records. Scientists, therefore, treat the mutation as a direct link between the physical remains, the recorded symptoms, and an accurate medical diagnosis. Finding a named mutation in the Romanov material has wider scientific value. It shows that modern genetic methods can recover medically meaningful changes from very old and degraded samples. The investigators combined careful field sampling, strict laboratory protocols, and methodical replication to read a single gene to clinical quality despite the age of the remains. Researchers compared the detected change to databases of known mutations to gauge whether it was a common harmless variant or a rare disease-causing change. The splice site alteration proved rare and consistent with disease reports. That assessment, together with repeated lab confirmation and clinical fit, supports the conclusion that the heir had hemophilia B caused by the F9 splice site mutation. Multiple research centers verified the finding. Teams at the Forensic Science Service in the United Kingdom, the Armed Forces DNA Identification Laboratory in the United States, and Russian research institutes ran tests on shared material. Scientists published methods and data in peer-reviewed journals. Later investigators applied newer sequencing technology to the same gene and confirmed the key result. The cross-checking by separate labs strengthened confidence in the genetic diagnosis. Reports remain available publicly if anyone wants to go through them. But even with undeniable proof in hand, one mystery refused to die. Could the story of the Romanovs still be incomplete? The unresolved questions. Even after many studies, some questions still linger. The most visible of these involved a woman who called herself Anna Anderson. She said she was Grand Duchess Anastasia. For many years, people doubted her claim. Laboratories later tested tissue and hair linked to Anna Anderson. The genetic results did not match the Romanov maternal line. Instead, the profile matched relatives of a Polish woman named Franziska Szankowska. That result removed the most famous claim that a Romanov daughter survived. Researchers also gave heavy attention to how samples had been handled over time. The earliest recoveries and records came from a period when keeping detailed archives was rare. The Soviet authorities kept many files closed, and some paperwork stayed incomplete. After the fall of the Soviet Union, investigators worked to create a clear chain of custody. Files created from the fields, images, maps, and labels recorded where each sample came from. Laboratories logged every transfer, who opened each container, and when each item moved between storage locations. Laboratory limits in the first round of tests caused debate. Tests in the 90s read short stretches of DNA and relied on strict contamination controls. Some people argued that long storage and early handling might have introduced errors. Scientists answered by repeating extractions and sharing samples with independent centers for verification. New sequencing methods became available later. These modern methods can read many short fragments at once and recover more of the nuclear DNA profile from degraded material. Some technical disagreements remain among specialists. A central point concerns which the daughter matched the small set of female fragments found near the main grave. Anthropologists compared bone measurements, tooth development, and growth plates to estimate age and sex. The metrics showed overlap for Maria and for Anastasia. The genetic profile placed the fragments inside the immediate family and eliminated outside candidates. Some experts wrote that the skeletal measures better fit Maria. Other analysts argued that the overlap left room for Anastasia as an alternative. Official and public reactions changed over time. On the 17th of July, 1998, Russian officials arranged a public burial at the Peter and Paul Cathedral in St. Petersburg for remains identified as Nicholas II and Alexandra Fyodorovna. The ceremony included relatives, government representatives, and religious leaders. The Russian Orthodox Church at first expressed reservations about whether all necessary tests had been run before burial. Later, after additional study and new tests, church leaders agreed to formal memorials and services. Scientists worked to make the record transparent. They published the methods used and data found so that other specialists could check out the results. Private laboratories in the United Kingdom, the United States, and Russia ran confirmatory tests. This cross-checking reduced the chance that a single error would change the overall conclusion. Research reports included detailed methods, controls used to spot contamination, and repeat runs that confirmed critical sequences. Institutions preserved samples and records so that later researchers could re-examine the materials. Museum curators, laboratory archivists, and state repositories keep teeth, bone fragments, and small artifacts. Each item in an archive carries documentation about where it was found and which tests were used on it. That practice allowed later researchers to apply newer laboratory tools to the same material. New studies used high-throughput sequencing and recovered additional nuclear markers that earlier methods could not reach. Legal, archival, and prosecutorial records grew into a public file. Investigators collected witness statements, police reports, and forensic summaries. Prosecutors and archivists compiled these documents so courts and officials could review what had happened. The assembled files helped government agencies and historians decide on burial, on memorials, and on public statements about the case. The files also supported requests to open archives and to allow qualified researchers to study samples. International collaboration proved crucial. The Forensic Science Service in the United Kingdom, the Armed Forces DNA Identification Laboratory in the United States, The Engelhardt Institute of Molecular Biology in Russia and other research centers shared samples and compared data. Private confirmation by multiple centers increased trust in the findings. Investigators coordinated methods and compared raw results to ensure that one laboratory did not rely on a single unverified test. The major scientific conclusions rest on several independent lines of evidence. Archaeology supplied context and artifacts dated to the early 20th century. Anthropology gave age estimates and trauma analysis from bones. Genetics provided repeated matches between the remains and living relatives. Together, these sources form a coherent identification that most professional observers accept. Minor technical debates remain in specialist literature, but they do not change the central results. In 2009, Evgeny Rogev and colleagues published a study that identified a mutation in the F9 gene in Romanov material. That finding connected the heir's medical records with a specific genetic change consistent with hemophilia B. Multiple laboratories later repeated key genetic tests and confirmed the result. Samples and reports remain available in scientific archives for qualified teams to study further. Researchers continue to keep materials available for reanalysis. New laboratory tools may recover further data from the same samples. Archives remain open to qualified teams who follow strict protocols for sampling and documentation. Science in this case advances in clear steps, with each method offering extra confirmation of earlier findings. The Romanov case now stands as a detailed record of research, laboratory examination, public review, and ongoing scientific study. Scholars and officials now update official records when new evidence appears. Even after a century, the Romanovs' story still whispers through labs and archives. Each new test brings new answers and more questions. Could more secrets lie preserved in the bones, waiting for future science to uncover? Tell us, do you think the Romanov mystery is truly over, or only just beginning?